Um, Hi all. Thank you for coming for the event, BioPython Working with Sequences. I am Pradeep, the president of the Bioinformatics Club. We have Neil, was the vice president, and we have Dwight Hall, was the secretary of the club. And right now, also we wanted to say that I shared the YouTube page for our club right now in the chat. Uh, just check it out if it's accessible. The previous event that we had on COVID Talks is uploaded there. And if you guys have any questions regarding that, you guys could shoot me, Dwight, Neil, any one of us a mail, and we'd be happy to respond. And so having said that, so I'm gonna jump in like a really, really short PowerPoint, just like, and then we jump into the hands-on exercises. Feel free to follow and like copy. I'm gonna share a Google Colab notebook. So feel free to copy that notebook to your drive and use it as a reference in the future. All right, let me just start the session. So, yeah, oops. I hope everyone can see my screen. So, the event today we're going to be is about like BioPython and working with the sequences. So let me just go ahead. So what we're going to do for this very short workshop is we're going to first go ahead and create our own NCBI profile and account. If you guys already have it, well and awesome. If not, it's really important being a part of the biomedical informatics field where we use a lot of biological data, and NCBI being the main database containing all the data, and having a profile or an account in it will give you like easy access for all those data points. And I'll tell you like how it actually works. And then we'll jump into like basic BioPython functions. So if you do not have any idea about Python or programming at all, it's cool because I'm gonna give you like a really short introduction on like what are the data structures that are present in the basic data structures that's present in Python and like the basic loops. I'm not gonna go deep into those stuff. It's like really extreme coding. We just need to know like, hey, so if you use this function, you're gonna get this biological pathway or method done. And if you have any doubts, like really like whatever doubts, if you even have it for the basic Python, you can always like interrupt me and ask me about it. And the third one was I had like a lot of people ask me about like, will the club help me? I mean, help them with project ideas and stuff. So I have like added a few resources at the bottom of the page. I mean, on the Colab notebook, so that you could like use it for your project ideas or like in the future, or you can always shoot us a mail, like because we like to like we like I'm a PhD student and Neil's kind of like a master student already, and Dwight is also a master converted to PhD student. So I've looked at more bioinformatics research, and if you have any doubts and like if you have an idea but you don't know what to do, you can always shoot us a mail and we'll be happy to respond to you guys about it. And all right, so the first thing would be for us to do is to create an NCBI account. And you can use that, I mean, you can use your regular Google account to do that. And Indiana University has a research account, which enables you to get more data. And it also, like, you can get like restricted data too. Like sometimes that's not available to public because it is only accessible to IU researchers. And it's really important because if you're working with 1 or 2 sequences, that's pretty fine. But then if you're working with like 10, 20 sequences, or like not 10, 20, like let's say 100 to 200 sequences at a time, then you don't want to like go to your web browser and open the NCBI account, download the FASTA sequence every single time. Rather than that, you could just do it within like 2-3 lines of code in BioPython, and that's where creating an NCBI account comes into play. I could go ahead and copy paste this. Yeah, I'll just go through the next few slides and then we will go directly into the hands-on, jump into the hands-on exercises. Well, what's BioPython? So Well, Python is right now one of the most famous and most commonly used language. Like literally every bioinformatics internship or job application requires you to know Python. And BioPython is the heart of bioinformatics in Python. So they have like multiple modules and packages built inside this big module called BioPython that would enable you to do basic biological functions. Let's say you want to transcribe a DNA sequence into RNA, or you want to like reverse transcript, like all these biological, like the central dogma. That's what we're going to be concentrating on today. Uh, uh, I mean, I hope all of you guys have taken like basic biology courses to know about the central dogma of molecular biology. If not, I'll just give you a brief introduction in the next slide. So it'll be like, I mean, you guys will understand it pretty quick. And so we also have an open resource called BioPython Cookbook, which I've also added in the note in the collab notebook. So it actually is a huge PDF document which tells you how to use each function and what resource. So basically just do a control F and say, hey, I want to convert this DNA to protein. You could just like check DNA to protein, and then it's going to land you on that page, and you could just use those functions. So that's pretty useful in a way. And then enters access. So basically, once you create an NCBI account, BioPython has this module called entries in it, which allows you to download as many data or like as many sequences or as many proteins or as many GEO files from NCBI directly into your system just via like two to three lines of code, which we'll be seeing right now. And finally, I just want to say, like, give a small explanation on what the central dogma of molecular biology is. So I'm hoping everyone knows this already, and I'll just blow past it pretty quick. So DNA gets converted to RNA via the method called transcription, and RNA gets converted to protein via a process called translation. And it has multiple steps involved in the middle, which it's a really elaborate method. I mean, if you guys want to talk more about it right now, you guys can jump in. This is not just me going to be given like a single lecture. It's going to be like a group discussion. Like Neil and Dwight and other members of the club, you guys can always jump in and tell what's about it. And the DNA replication is basically the DNA getting converted back. I mean, like replicating into like one strand of DNA becoming multiple strands of DNA. And so that there isn't really any bioinformatic analysis going down there. But then conversion of DNA to RNA, where you have to choose which strand of DNA is going to get converted into RNA. And also RNA has a different profile, as in DNA has ATGC, but then RNA has uracil instead of thymine. And also the translation, where codons, like you have a start codon and you have a stop codon, which will tell the processors, like the ribosomes and other molecular processors, that where the actual protein starts in the RNA and where the actual protein ends in the RNA. And it'll also give you like, hey, so if I see these ATG, I want to start the protein. If I see like ACC, I want it to be a cysteine molecule coming over there. And that's how proteins are made. I mean, I'm sorry I'm not able to go in depth because it's a really elaborate process. People take like 2-3 lectures to cover this whole dogma. But I'm hoping like most of you guys know it. If not, we'll go through it again when we go into the code. So let me just stop sharing a bit, and I will. So I'm going to share you guys my collab file. So feel free to go to the file option and copy it to your own drive rather than editing it in my like collab notebook directly. So let me just copy the link, and I'm going to put it on chat. I hope everyone has access to it. Just let me know if you do not. And NCBI. And I'm going to post another link, which is the NCBI. So the first thing we're going to do is go to that link and create our own account. So I'll just walk you guys through it. It's not a complex process, but we would want to know what is our key and the email ID that we can use. So just a second. All right, so let me just share my screen, and here we go. I hope everyone can look at my screen. So this is the newly updated NCBI page. I mean, this actually leads to PubMed directly. So, all right, so what we're gonna do is log in to the account or sign up. I mean, I don't want to log in with the Indiana University right now. It's again, you need to use your like, what's the thing called? The geosecurity and stuff. I don't have my mobile phone right now, but I'm gonna use a Google account right now. So yeah, some random account to so once you're logged in, you need to go to your dashboard. So I hope everyone has, I mean, so you probably need to type in a bit more details and stuff, but if you already have logged in before with your email ID, that'll be cool. Does anyone have any questions on this? I mean, it's pretty simple, but I'm just gonna wait till you guys completed it. All right, so I was gonna say, so the best part about having your own account is you have your own search histories. And so let's say you search for a particular package, a particular paper, like a particular topic that you wanted to work on, and you can always go back to that search and check out if there was new papers popping out or new data points popping out. And yes, this is basically like, these are my collections that I could save. So if I'm writing some paper or I'm on a project, I'm working on something, I could save them to a particular collection. And that collection keeps updating every time some other researcher adds on a paper or some data. So this would make it easier for me. So I can always come back and just look it up rather than me going through the same process of searching it on the search bar and then filtering it out based on years and stuff. So that is one big advantage of having your own account. So I'm gonna show you, yeah. So if you go and click on your email ID that's on top, you should be getting your NCBI account settings. So, or you could directly go to settings from NCBI, and your email ID and which needs confirmation, probably they'll mail you to your, they send a mail to your, yeah, they'll send you a mail and then you need to click the confirm button, and your API key. So when you're using BioPython, if you do not use an API key, you're only given like 3 requests per second. And it also has a restriction of about like 100 sequences at the max. But if you have an API key, you can like the limit moves up to 10 requests per second, and you do not have any limits. So yeah, so I hope everyone has come to this step. If you guys have, I'm just gonna like pause a bit, and if you guys have come to this step, please do a thumbs up on the participants, so I could like if you guys have any questions or something, I could just look it up. All right. I mean, is anyone stuck anywhere in this signing up process?
Hi, I asked a question in the chat, like how do I create that API key? I just click on create an API key?
Yeah, just click on create API key, and you should be getting yeah, your own API key. My bad, mine got like created automatically. Yeah.
And what is the advantage of doing this? Can you repeat that again?
Yeah, so so when you're using BioPython, which we're going to be doing, like the right next step is when you do not have an API key, you can work on like, so it actually, the NCBI allows you to like obtain or download 3 sequences or like 3 proteins per second, and it has a maximum limit of 100. So you can't really analyze a lot of data points. Like being in bioinformatics, you will always find yourself in situations where you need to like download like 1,000 sequences, or like if you want to download like all the variants in the genome and stuff, it's going to take a long time for you to download it, and you need to send multiple requests. But if you have an API key, it means you're a trusted user. So it automatically, like you don't have any limitations on how many sequences you can download, but it asks a 10 requests per second. But if you have a research account, which uses from Indiana University, it's just way more faster.
Okay,
So
thank you.
yeah, no problem. All right, so just keep this tab open because you might need to copy paste the API key. So, all right, so let's jump into our hands-on BioPython part. Can you guys show me like give a show of hands, like how many of you have done Python before? So I would know if I need to. All right. Awesome. All right, that's cool. So, all right. Most of you guys have done it. Yeah, if you guys have any questions, yeah, you could go ahead and unmute it and let me know. Well, I would just go through the basics again, but if anyone has any doubts, feel free to interrupt me and ask about it. All right, so Google Colab is a really easy and effortless way for, like at least for us, when we are beginning to learn Python or beginning to install, like work with new packages. So yeah, awesome. Yeah, I'll go through the introductions too. I'll just make it like pretty simple, but you can always like email me back, and we'll also have a separate session too. So all right, so when you're using your like in a Colab notebook, it's gonna be pretty simple because you don't have to worry about downloading the packages in your system. It's gonna just download it on the Google Cloud, and everything is based on cloud, so you don't have to worry about any setting issues or anything else. So it's basically like Jupyter Notebook. You just type in your code over here. If you need a new code block, there's a plus code over there. And then you all need to do is like click on it, and it's going to give you a new code block. And you could type, let's say, a very simple Python command is printing it out. So you could just do print, let's say, AB, and you could just run it, press it on the run button, and it's going to run the whole command. It's going to give you the output at the bottom. So it's a pretty simple notebook to use, and it's not complicated at all. And if you want to install, so what we're going to do is first thing is to install the BioPython module. So it's a pretty simple command. If you want to install any package in Python, this is the command to go. Oops. This is the command to use. So pip, which is another package in Python, which should always be present. So it's like an inbuilt package that comes along with Python. So you could use pip install and the package name. So if you go ahead and run this cell which says pip install BioPython, you should be, because I've already run it, it says requirement already satisfied, but you guys should see a process bar running through and it downloading. So that means your BioPython is installed into your workspace. All right, so when you want to install a particular package inside the whole module, so don't be confused because BioPython is the package or the module's name, but then bio is what we call in Python. It's because the name's pretty long and they wanted to make it short, which is pretty ridiculous. So you could do import bio, which is basically importing the whole BioPython, but Python as is really, but it's not the best part of Python. So if you want to import a particular small module in it, you need to specify, you need to like re-import that specific module. So today we're going to only talk about sequences. So we're going to import the seq package from bio. So this is how we're going to say. It's pretty common, like just like talking to the system, like, hey, from this module, import this package. So that's what we're going to say. So from bio.seq, import seq. So this is a very common command. And this is usually given, if you just Google BioPython, you should be getting like, hey, this is how you install us, and this is how you use us. So this is going to be like a very standard command where are we going to use. If you're going to, let's say, you're going to import another package called numpy or pandas, you're just going to say import numpy or import pandas. So, so the importing part is a pretty basic and most standard Python function. So that is how you import the seq package in BioPython. So I'm gonna like talk about the most common data structures that we at least gonna be using right now. So one is gonna be oh, okay. So I'm just answering a question on chat. So if you want to, so what you're gonna do is go to File and save a copy in Drive, and that's gonna save the whole collab file into your own drive. So you could go back to your. So once you open your drive, and you should be getting another new notebook that says copy of the original one. So here you could just go ahead and this is going to be saved into your drive, so you could just use it whenever you want to, and not just like if you edit it over here, it's not going to change in the main file. I hope that helps. Awesome. So, oh, I didn't see the time over there. But yeah. All right. So let me just go to the basic data structures that are present in Python. So one is a list. And to make a list, all you need to do, so list is basically a list of any type of data. I mean, like it could be it could be a bunch of numbers separated by comma. It could be a bunch of strings separated by comma. So basically, if you want to make a list, you put the square brackets in, and you add in whatever values you want to, like let's say 1, 2, 3, or a, 1, b. And you need to separate individual stuff with a comma. And that's basically a list. So lists are pretty useful when you iterate over multiple stuff, which we'll be discussing later. And let's say, so next thing is dictionary. So dictionaries are basically to label one part of, yeah, it's basically like, imagine you actually have a dictionary and you want to know the meaning of a word. So you go and look at that word and then you get the meaning. So the word is basically the key and the meaning is basically the value. So let's say, and it's represented in curly brackets over here. And you're going to say, hey, for this key, I want this value. And it's going to be separated by commas again. So basically you can say, well, if you want to like make a dictionary of like let's say sequence, like purins and pyrimidines, like AT and GC are much more easy. Let's just say coffee and tea. Maybe like okay, I want mocha, which is a coffee. I want chai, which is a tea. So that is basically a dictionary. So let's say like if you're gonna make a new dictionary called cafe, you're gonna use the curly brackets, and you're gonna be like, hey, the key is gonna be coffee, and what type of coffee do we have? And it's gonna be let's say a mocha. So something that's similar. And then we have strings, which is like large sequences, and then we got tuples. Strings are basically just the whole lines, or like just the whole stuff. And then you got tuples, which are represented in regular brackets. The thing is, tuples, you can't change them in the future. And say, okay. And then we got, so if you want to work with these structures, the strings, you got to do dot lower and dot upper. So basically, dot lower changes the whole sequence in lower cases, and dot upper converts the whole thing into uppercase. And so I've actually given complete comments for the basics over here, and you guys could just run the whole cell, and it's going to give you the results. Like, let's say if this you assigned this to stat. Well, to answer Anna's question, yeah. So if you have different cases, then it'll give you errors when you're converting into proteins. So that's why we use either lower cases all or just upper cases on all. And yeah, so that's a good question. So I've actually, the whole cell is pretty self-explanatory. And if you want to print a variable, so these are all variables that we just declared over here. If you want to print a variable, you're going to use the F command inside the print. So if you want to print just regular print, you could just do this. Just print regular brackets and then inside quotes, whatever you want to say. If you want to print a particular variable inside, you're going to do F before you put another bracket in. And then inside double quotes, you could have a curly bracket in the variable's name. So variable stat underscore lower is this. And then you have like the curly brackets and stat underscore lower. So I'm going to skip the for the loops over here because I think we're already running out of time. We haven't even jumped into the BioPython part yet. My bad, but all right. So this is what I'm talking about, where we're going to, so here you could enter your email ID and then your API key. So what we're going to do, and before that, you got to import entris package. Same thing that we did with the seq package, but then with the entris package. And once you import it, once you run this cell, it means your Python notebook is linked with your NCBI account, so you can easily access all the data from there. And so I'm going to be starting with the sequence objects now. So sequence objects are basically string-like objects, but what they have is just the basic command called seq, and then brackets, and then whatever you goes inside, like ATGC. So because it's going to be DNA sequences, it's going to be AT and GNCs. But if it's like something else, RNA, it's going to be a U and GNC and more. And you can even have your own sequences. Like, let's say there was X and Y sequence in bacteria like a few years back when the scientists discovered that recently. But yeah, so let's get up to the most basic thing that we could do is count the number of occurrences of a particular. Let's say you have like a huge sequence and you want to say, hey, I heard this AA's actually influence this disease. I want to see how many AA's are in the sequence. So for that, you could just use a very simple command that's called .count. So let's say you have a sequence. I'm just going to copy paste this over here. So you could assign a variable like seq equals to capital SEQ, which means it's a sequence object. And then inside brackets, you could just type in the name. And then if you want to count how many occurrences of this, whatever it could be, like you could just do a single A or a single or double A or triple A. How many occurrences of that is present in my variable seq? And that's going to give you like how many occurrences it has. That's a pretty simple function. And the other function we have is the GC percentage. So basically how they look at the DNA sequence and they tell you if it's a good DNA sequence or not so great DNA sequence based on the GC percentage. The GC percentage is basically how much G and C nucleotides are there on the whole sequence. So basically we have AT and G and C, which means you should have at least 50% of the whole sequence to be G and Cs if the whole sequence is at random, right? So if it's at 50 and slightly below 50, that would mean it's a good sequence. If it's like about 60% and 80%, which means it's not so great because it will mean like towards towards the end of chromosomes, you get a lot of ATs. That would mean it's not really important. So GC is a pretty easy function. You could actually use like multiple loops and stuff, but then it's a very simple function over here. You just need to import it again from this pack. And bio is the main package. And then you call something called cqtils, and you import the function called GC, and then you do the function. So you could just basically run it. And slicing is pretty simple. Slicing is basically when you want a particular area of the whole sequence. So let's say you want the first 10 nucleotides. So basically, you could just use square brackets of that and then just say 0 is to 10. And it'll give you a sequence objects containing the first 10 nucleotides. So also a very important thing to remember if you're in programming and if you're using multiple languages, or even if it's beginning, Python starts from 0. 0 is basically 1 in regular world, but in Python it's 0. It's like everything starts from 0. So you can't basically say 1 to 10, it's going to be 0 to 10. This is actually, the G actually is in the 0th position. So that's a really important thing to remember, because like even I make like a lot of mistakes, even though I've been working in Python for like 3 years, I still make a lot of mistakes thinking like it starts from 1. And if you want to reverse the whole sequence, there's a pretty simple trick. Same thing, have you like do slicing, but then you introduce -1 towards the end, and it reverses the whole sequence out. You could, if you have any doubts in that, you could let me know. It's a very simple trick, which is used, asked in a lot of interviews, coding interviews and stuff, because people try to build their own functions and like a huge code block, but then it just can be done pretty simply over here. Yes, and then concatenating 2 sequences. Basically, adding up 2 sequences is pretty simple in Python. You just need to use something called a function that is called join. So join just like adds up 2-3 strings together based on what's inside in the middle. And there's another function that we have in so yeah. So right now we'll not worry about this because I haven't told you guys about for loops yet. But so let's say you have like sequence A and sequence B and sequence C, which is in a list over here, and you want to, and let's say you want to connect them, but then you want to connect them with 4 A's between them. So you want to be like ATG and AAA, and then ATG, ATCCCG, and then again 4 A's, and then TTGCA. So if you want it that way, you could say a spacer or some other variable that's called this sequence, and then you could say spacer dot join, and then your list of strings or sequences. And when you run it, yeah, so you could see specifically it's going to merge between multiple sequences with 4 A's. Yeah. And all right, so the next thing we're going to do is a fundamental biological process, which is reverse complementation. So you have a regular DNA string. You want to find out what its paired up string, or like the other, like if you have a template string, you want to see what the other string is. You can use a very simple command that's called comp.complement. So if you have your sequence, that is called seq of the whole. Like you might get this from some data, and you want to find out what is the reverse complementation, as in what sequence goes against this. You could just do myseq.complement, and it'll print out the reverse. And I've also given you guys functions over here, reverse complement, for you guys to test it out by yourself if you're interested. Reverse complement basically reverses the whole sequence and complements it. So yeah, feel free to edit it in your own collab notebook and test it out. All right, so let me just go to, yeah. Okay, we have, all right. I'm just checking at the time because I think I'm taking a long time. Is that Neil? Like, do we have more time yet?
I feel like a lot of people have classes and everything, so try to.
Yeah, I'll just move a bit fast, but this is going to be posted on YouTube, and if you guys have any questions, feel free to like mail me too. I'll just go through it pretty fast. It's going to be like pretty simple functions. So when you're like moving in, let's say you want to convert the DNA sequence into RNA sequence. So let's say we have my sequence, which is the sequence variable of this DNA, right? And you want to convert that into RNA. There's a very simple function that's called .transcribe. That's all. And you get your own RNA sequence. If you want to understand the process, you could just look up online, or we could explain it to you via mail or something. And if you want to do a so if you want to transcribe, you want to find the mRNA sequence that goes along with this sequence, like as in the other strand of the DNA, and you want to find the mRNA sequence of that strand, you could use a chain of functions like .complement and .transcribe into one single tool. Like you don't have to do it multiple times. Like you could also do this like this. So instead of doing 2 steps, you could just do it in one single step. So yeah, you guys can, yeah, don't worry about it. Don't worry about like leaving right now. This is gonna be on YouTube, so feel free to subscribe to the channel, and we also post a collab notebook over there. All right, so the next one. So once you run through this, we could have the translation, which is basically converting your mRNA into protein. So this is a really important process, because you can actually change the organism that you want to convert the mRNA from protein to. You can either choose a bacteria, which has a different process, a human, which is a different process, and let's say a virus has virus kind of imitates human process, but also might have different processes. So you can use the bacterial table or the human table for now. So table 1 basically chooses the mitochondrial, that is basically the human table. And table 2 chooses the bacterial table for converting, bacterial process for converting your RNA into protein. So all you need to do is do .translate if you just want to do it in humans. If you want to do it in bacteria, you're going to specify, hey, it's going to be table equals to 2. Or you could just say table equals to bacterial. Both of them work. And as I said before, you have a lot of stop codons, which means that's where the process will stop. But that's where the protein is going to stop. But if you want to keep looking, if you want to keep like, hey, just go ahead and I want to see what sequence is after the stop codon 2, you could say 2_stop equals to false. But if you actually don't want to do that, you could say 2_stop equals to true. And if you want to change the stop symbol from an asterisk into something else that you want to customize it by yourself, you could change it to whatever you want, and it should work. So that's basically for your own representation. So, and if you want to, I could actually skip over this part because I need to tell a lot about like, hey, what is this table about? I think we'll do this along with the next session of BioPython when you talk about proteins more. But most of you guys who already know what a codon table is, feel free to look it up. So one is going to be the standard table, which is the most complex, like a common table. And then we have the mitochondrial table too. And all right, so I'm going to go over here and tell you guys like how to import sequences from NCBI, which is one of the key things that we want to discuss here. So you're going to import something called entres, which is again a package in bio. And then you're going to again enter your own entres email ID from the NCBI account and your, let's say, I'm going to go ahead and do that actually now. Oops. And your API key, which is pretty important too, because you have right now we're going to try and download a lot more sequences. So I'm just going to go here and say API key. Hey. And so what I'm going to do is the IDs is basically a list of nucleotide IDs that I've taken from NCBI. and I'm gonna like go through each of these IDs, get their nucleotide FASTA sequences, and I'm gonna record, and I'm gonna make them, append them into this list called record. So it's completely, oh, what's up? All right, so what we're gonna do over here is use the function called efetch, which fetches the data from the nucleotide database and has the ID of like, okay, fine, I want, these are basically sequence IDs. So you're gonna refer like, okay, fine, I have sequence IDs and I want you to get the sequences from it and what type of sequences and what type of text, I mean, what type of file you want it in. If you can either have it as a like a regular sequence file or like a text file. So and record is basically an empty list over here, which we're gonna keep adding data using a simple function called append. It basically keeps adding to the list. And once you run it, I'm hoping, oops, what happened here? Oh, all right, I think my breakfast isn't working here. Scratch that. Oops. I think we'll go back to this part of code in the next lecture series because I think there's some sort of error in the end. I'll look it up again and we will come back in the next lecture series that we're gonna do on. But yeah, and then to wrap it up, we have the resources that's available. So I have like multiple GitHub pages and a BioPython tutorial HTML. Feel free to look it up. And this is where I got most of my resources from. Yeah, so you could just basically, this, if you see, you have like 24 chapters worth of BioPython tutorials, and it's always updated. Like last update was 2020 in September 4. So there's like multiple versions, and it always keeps adding new functions and stuff. And you have multiple, if you want project ideas, look over here, like which has like how to use like tools and resources. And you also have like, if you want to get started with Python, they have this amazing Google notebook, which is on 100 days of Python. So from the basics, you want to like work on multiple projects, and then you'll be pretty strong by like day 20 itself. So yeah, so that's about it for all right now. Thank you for coming to the event. We're going to post this on YouTube, and feel free to ask questions anytime you guys want to. I'm sorry I couldn't like cover everything together because of the time crunch. Next time I'm going to plan it much better, and hope to see you guys in the next event too. Neil, if you want to